green signal on the chat or something, and then
Go
now
ahead.
it's
Yeah.
we are live.
Yes, yes.
Okay, perfect. So, welcome everyone today for the podcast with our series, The Next Experiment. And today we have Professor L.S. Shashidhara as one of the guests on the platform. And all of this is also due to our audience and also the team who is especially working behind to make this project successful. So without giving more moments, I would really love to start with the introduction of Professor L.S. Shashidhara. So today we are privileged to host a scientist whose work has influenced not only modern biology but also direction of science education and research leadership in India. So Professor Ellis is a distinguished biologist specializing in genetics, molecular biology, and evolutionary biology. Over the course of his career, he and his group have uncovered key genetic mechanisms that control how organs develop, how their size, shape are determined, and also how growth is regulated during the development. While much of his work has been done using fruit flies, Drosophila melanogaster as a model system, its implication extend to understanding how human diseases and also epithelial cancers arise. His lab has also contributed to functional genomics and the development of fly models to study many human diseases. But beyond his scientific discoveries, Professor Ellis has also major force in shaping the scientific landscape of India. He is currently the director of National Center for Biological Sciences (NCBS) in Bangalore, which is one of the India's premier research institute in life science. He is also a professor in ISER Pune and also holds a honorary and visiting professorship in Ashoka University. Over the years, he has also held key academic and leadership roles at major institutions and contributed significantly in the research ecosystem, mentoring young scientists and also strengthening interdisciplinary science in the country. His contributions have been recognized, and some of the key honors of Indian science. He is a recipient of Shanti Swarup Bhatnagar Prize in Biological Sciences. He has also received J.C. Bose National Fellowship. He's an also elected fellow of Indian Academy of Sciences, the National Science Academy, and also National Academy of Sciences in India. He was also elected as an associate member of European Molecular Biology Organization, EMBO, recognizes his global achievements and also standings. Today, he also holds a lot of directorships and also key roles in facing in empowering students. We truly set him apart is the commitment in science outreach and education, especially reform and connecting science with society, which resonates strongly what we also do over here. We welcome you, sir, today for the
Thank you. Thank you.
podcast.
I'm really honored to be here. Thanks for the opportunity.
Thank you so much, sir. So, before moving into very research oriented questions, we would love to really drive this conversation in a bit candid way. So, as a classical scientist, or me being a classical podcaster, this is a key question that I ask, and also we would love to hear it from you. That as in childhood, what made you motivated to pursue science?
Okay.
And why actually
Yeah,
biology?
yeah, sure, sure. So there are two aspects to what you say pursuit of science. One is reading about science or scientific discoveries. Second is actually, thinking of that. I also want to get into research, right? Scientific research. So the second part that I also wanted to do research came much later, and not as a child, not as a school student, not even as an undergraduate student. So I grew up in a family of academic people. My father also was an engineer working, but he was really an academic person. And my mother and my aunts, uncles, everybody used to read a lot, particularly literature. And and the question was always the questions rather in the family, in all our discussions were like on the three things: how, what, and why, right? And no one accepted anything if you say, unless they get satisfactory answers on how, what, and why. Very argumentative family, I can say. Then, in fact, I wanted to study mostly history and literature for a very long time, until my 10th grade. I used to read a lot of those literature from around the world, as well as some philosophy books of Radhakrishnan and others. But later, when I moved to undergraduate program, coming again from agriculture, family background, farmers background, agriculture sciences really attracted. So I joined BSc Agriculture Sciences in Dharwad, which is also a place for culture and literature. That also influenced me a lot. And in during my undergraduate studies, some of the experiment they explained of in agriculture, some very fundamental experiment you can call now, but those days perhaps it looked as if they are trying to do something completely out of the box. Like simply, for example, how much influence of soil on plant growth. Even such fundamental questions were empirically tested. Or can we hybridize cabbage and carrot and make cabbage on the over the ground and carrot under the ground? These are the kind of questions people asked. Of course, most of the experiments actually failed in those days, but actually they started thinking in that direction. That's what made me to think that, okay, research is the most exciting part. It's not about success of doing of any experiment. It is the very process of doing experiments, the way you design an experiment, and that's
Mm.
what I really fascinated. Even today, for me, designing an experiment is more exciting than actually doing it or worrying about what results it will come out.
Mm-hmm. Do you still design experiments for your students sometimes?
Of course, I leave it to my students more often than earlier, but um,
Mm-hmm.
but I still like discussing with them their design of experiments, because it's not correct
Mm-hmm.
for me to design and give them what I think
Yeah.
that I should expect.
Mm-hmm. Mm-hmm. So you mentioned your journey as a child, and once you entered the college. Uh, so when did you decide in that process that now, okay, I want to pursue research or PhD, and also you decided that okay, I want to move abroad, and I would look for opportunities or.
Yeah. So in my fourth year undergraduate program, I got BSc Agri with a 4-year degree course. In the
Mm-hmm.
fourth year, we were supposed to do small research projects, and that
Mm-hmm.
um, um, so in a way to um, what I'm saying is sort of getting really into research myself, right? And that's when I
Mm-hmm.
decided that I'm going to do PhD too. And then obviously you have to do masters on the way to do PhD. So I did my PA, my masters also. I did my through research, I published papers. But research, I
Mm-hmm.
actually applied to a couple of places. Finally, I got into CCMB in Hyderabad, Center for Cellular and Molecular Biology. I just entered CCMB and was going through some coursework. Then when an opportunity came, completely, you can say accidental way, I applied and got it. And then, in doing PhD in Cambridge, along with full scholarship. Then I discontinued from CCMB and went to Cambridge to do my PhD. So, and then of course, once you enter into PhD and you knew the rest of your life is going to be a research-based career.
Absolutely, that's completely true. So, how different was your PhD and postdoc experience, if you recall, and go back in the time?
So okay. Let me first explain PhD. So I started as a plant biologist, as you know, because of agriculture
Mm-hmm.
science. Then my
Mm-hmm.
PhD was also in plant sciences. But during my undergraduate and postgraduate studies, a lot of my courses are all on classical genetics, cytogenetics, as well as evolution. Right? Without these three, you can't do good plant breeding and genetics. So if you want to develop a new varieties, if you look at the works of Ms. Swaminathan and such fantastic plant breeders, you actually, identify the foundational concepts of genetics, cytogenetics, and those are genetics now we call simply genomics, because we have much easier to do genomics than working on cytogenetics, and then looking at linkages and all kinds of things. So identify, design how you develop a new variety by crossing, distant, lines and so forth. And during that time, I they always used to give the examples from Drosophila biology and how fundamental concepts were established. In fact, the very origin of cytogenetics, we go back to the Columbia fly room, and when Thomas and Morgan and students started looking at the chromosomal basis of heredity. Similarly, much of evolutionary studies also people have used Drosophila as a model system. Muller, Dobzhansky, and many others. So given that, it's really attractive. So my PhD was for 3 years in UK. Typically in those days, it is 3 to 4 years. Thankfully, I could finish in 3 years. I immediately jumped on to do fly biology, Drosophila biology, because that's what had already, imprinted in my mind that, that's what really I like to do more. And then I did a very short postdoctoral for about 15 months. Then I knew at that time, me and my wife, that we could to go back to India as early as possible. And then opportunity came up, and then I came back to India in just within no time. So my postdoctoral stint was very short, but that gave me the transition from plant biology to the Drosophila genetics and Drosophila biology.
I see. So postdoctoral experience actually shaped what then you started working as part of
In a way,
your
because the
independent
concepts are not
research.
very different. By the time
Mm,
the way
mm,
our teachers taught me in undergraduate, postgraduate time, and also I
mm,
used to
mm,
attend a large number of seminars in Cambridge. Cambridge is a hotbed for these
mm,
good seminars. So I used to attend large number of seminars outside my field too. That helped me to understand that its foundational concepts are same. It doesn't
mm,
matter
mm,
work. But what I wanted to learn at that time, or needed to learn, was only certain techniques which are Drosophila specific. But I don't
mm,
have to be in my genetics or evolutionary biology or this one to get into that work. I just had to learn few
mhm,
specific techniques, but the rest all is the same foundational concept which I already learned. So that was a
mm.
smooth transition.
I see. So do you sometimes recall any joyful, funny, or something like those incidents
Oh
during
yeah,
your
yeah.
PhDs and
Yeah,
postdocs?
I tell you what. There are of course quite a few. The one particular
Mm-hmm,
one, uh,
mm-hmm,
the two I can tell you right away. One is at the end of my PhD, and those days
mm-hmm,
it was you hand, you had to do it manually, and then you have to run a long gel, and then you manually read the sequences, dot by dot, right? And
mm-hmm,
aligning the sequences to what is already known, what the sequence tells you, is a really a lot of work. And
mm-hmm,
sometimes the people would spend a whole PhD in just sequencing a small
mm-hmm,
piece of DNA. So I sequenced about 3 kb DNA, and so I wanted to do some analysis. George Church
mm-hmm,
was just at that time had just started as in independent work in genetics department of
mm-hmm,
Cambridge. He organized a
mm-hmm,
first workshop on using computers for sequence analysis. And it was just the beginning. There was no online sources or anything. There was no internet in
mm-hmm,
1991, right? So I actually, registered for that, and that helped me to quickly learn using computers to do
mm-hmm,
sequence analysis. And as soon as I might join
mm-hmm,
my postdoctoral lab, my postdoc supervisor told me,
mm-hmm,
since you come from molecular biology background, we are all geneticists. Why don't you do the molecular biology part, do the sequencing analysis, everything? And he told me this is the gene we have identified a certain function. You want to know what this gene is. So I
mm-hmm,
quickly looked at the sequence and analyzed using the computer. Within 3 months, found out that particular, what is the nature of that gene and everything. And within 6 months, we got a paper out of it. And that project got closed, and I had to restart some new project. What I thought that I will do,
mm-hmm,
because the computer is got really accelerated. The
Mm-hmm.
second interesting is, particularly my experience, which I can never forget, is when I found
Mm-hmm.
first transgenic fly when I was doing generating transgenic flies. So we keep on injecting. Those days, the injection systems were not sophisticated. To generate transgenic flies, you have to inject the DNA that you want to insert into the genome of Drosophila through in the embryo. You would see, inject hundreds of these things. Sometimes they don't lay embryos. You have to inject within 1 hour of, after laying them, fertilization in a way. So during that time, you have to sometimes work the whole night, and then all are white-eyed. You have to get a red-eyed fly in the population of the white-eyed flies which you have injected, and then you keep on looking at larvae, you keep on looking at flies, and suddenly I found one, right, a red-eyed one. It was such a joy, and then a whole night and a couple of days, I couldn't sleep well. Whenever I close my eyes, I would see one red-eyed fly, the ocean of white-eyed flies. So it's
That's fantastic.
yeah, yeah, it's a fantastic experience.
That's super nice. So you mentioned, quite well that you were excited, and being a young researcher at that time, this would be super nice and joyful. So what do you think, in student in general, how important is curiosity as compared to talent when it comes to becoming a good scientist?
Curiosity, of course. Talent is something which is very subjective. How do you want to define talent,
Mm-hmm.
right? Sometimes if someone has a lot of memory and keep my telling whatever you ask the question, and they come out from whatever they have read, and you call it as a talent. It's simply because they may have
Mm-hmm.
memorized well, maybe have a very good memory, because memory is also some
Mm-hmm.
structural aspect of your brain, and
Mm-hmm.
and sometimes the way how you grew up in the family, there are certain sharpness to your memory and so forth. Second part of talent is basically analytical methods you employ.
Mm-hmm.
Also comes by training. More you do research, more analytical in your mind. So the talent can be
Mm-hmm.
built up, talent can be further developed and advanced. So in a way, talent is not something which is simply called it as a bond with talented kind of thing.
Mm-hmm.
So, but what you can, that's why in that context, to be part of a research group and continue to be doing research of high quality,
Mm-hmm.
not get
Mm-hmm.
bored. For decades, you want to research. The curiosity is the only way. If you get
Mm-hmm.
not curious to ask questions, you're not curious to find the answers, right? And then how will you pursue science for decades, right? If you're someone that
Absolutely.
started, 20, 22-year-old, and if they go up to
Mm-hmm.
75, 50 to 60-year-old
Mm-hmm.
research,
Mm-hmm.
you need to be always curious about things. Otherwise, you can't do good research. You may simply do research because you have lots of students, you tell them to do something, publish papers. It's a very
Mm-hmm.
job, which is a routine job kind of a thing. No, no, no. Research
Mm.
is not a routine job. Curiosity is the most important.
Definitely makes really, I think for students, this would be a really nice boost today, that, okay, what they should really consider before entering the domain. So I'm definitely sure that when you were a student, and today you being an independent researcher and holding several positions, things would have gone overwhelmed over the time, and they would start stressing at a point. So how you used to cope up when you were a student, versus now, what activities do you try to do which manages this overwhelming environment sometimes?
I don't. I mean, see, the thing is true. I mean, the compared to earlier days, today is much more crowded and overwhelming sometimes. But then you also have to look at from other perspective. The earlier days, you have fewer resources, fewer places to do research, and now there
Mhm.
are thousands of places to do research, thousands of different ways of doing research, thousand
Mhm.
different ways of accessing information. Earlier days,
Mhm.
I used to go to library and sit in the library, you want to get some
Mhm.
information. So during the experiment, you think of some question, then you have to wait for a day, you finish the day's work and then go to the library and find the answer. Sometimes you would forget the question that you asked, right? But in
Mhm.
today, in between 2 minutes, if you have time, you can just, voice your question to the Google and Google find the answer for you, right? Or PubMed, whatever.
Mhm.
So I think with the technology, with tools, with so many people in the community, doing science is fun. It's actually more fun than earlier, right? Earlier, it's your own driven curiosity will drive. Today, it's a collective curiosity. There are so many people, right? You will get
mm,
excited because in the coffee table, in this one, and wherever you go in the dining hall, there's so many
mm,
interesting conversation can happen. I think
mm,
every time has its own excitement, and today's time, I look back, I think is more exciting. I don't think people should think that it's overwhelming.
mm.
It's just more exciting,
I
basically. Because
I
science
see.
is still social endeavor. Because in science, what you do is to go beyond your own perception, beyond every human's perception. Otherwise, would not have
Mm,
discovered uh, is the Earth which moves around the sun. Because every individual ever lived on Earth or even living today, 8 billion people would see the same thing, sun moving around us. Then how do you know what is true, right? So science has provided this. Science has enabled to see the
mm,
world or naked eye cannot see, whether it's the size
mm,
or the and the sound everywhere, right? Here. So
mm,
given this, science has to be a social endeavor. You have to make it a
mm,
community. It cannot be one
mm,
individual sitting under the tree, right?
mm,
So and that collective endeavor is easier now with so many people working, easy to connect people on like what we are doing
mm,
now, right?
mm.
Across. And so it's really exciting rather than thinking that it's overwhelming.
I see. Absolutely. So you nicely mentioned, the shift and you being a conjunction today, in the times when there used to be no PC or almost getting newly freshly started versus now where you see everything moving fast forward in the science with AI and tech and everything, that sequencing results are like ready within a day. You can analyze the whole data. How do you cope up with this growing evolving science, and how do you keep yourself updated and adjust according to the current
I mean,
generation?
no. So each individual have to find their own ways. I'll tell you what personally, I'm
Mm,
not going to be prescriptive. I don't want to, recommend one
mhm,
particular way. Let me tell you what
mhm,
I. So obviously, you sort of delegate. You sort of network and you redistribute the task, right? For example, you
mm,
run a journal club in the lab. You don't have to go back to library and then try to read everything yourself. Others can read and tell you what it is, and then you can keep asking questions, get for clarifications, or you
mm,
can attend seminars, conferences, and kind of thing. These days, you can
mm,
also attend webinars online and so forth. Right? There is a
mm,
multiple ways of understanding things without you have to spend more time on it. And now, of course,
mm,
even those are all downloadable and so forth. The third is, you know, how you cope with the changes and kind of thing. Say, for example, in my case, whatever change happened with a very natural way and not because I consciously wanted to make changes. So
mhm,
when I was looking at the Drosophila wing and halter development, differential development of two organs of a fly,
mhm,
which
mhm,
homologous organs, and this led to asking questions: how come in other insects there are different way of body patterns are different than flies, right?
mm,
All insects are one type and lepidopterans are another type, and
mm,
tribolium.
mm,
Led to some evolutionary biology question, what you call as
mm,
evo questions. And
mm,
then and we had enough tools to look into it, right? You can do
mm,
genome
mm,
for other insects and you can collect information and then do cross, experiments and so forth. Then the COVID hit, right? And during COVID time, we thought we know so much about molecular biology. We know about what is DNA, RNA. We know how to do RT-PCR. Why don't we contribute to this? We also know about immune system, right? Why don't we contribute? Because this is the first time such a pandemic occurring after 1919. It's a global pandemic where we can actually use modern molecular biology understanding to address this problem. Right? Think of it. If there was no RT-PCR test available, how many people would have just moving around without thinking that they're carrying virus and spreading to others who are more vulnerable, right? So actually, it saved lives. Billions of people were saved because of RT-PCR tests that we had, and later, of course, vaccine. So that's why I got into infectious disease work, public health, and so forth. It's simply because, as a biologist, I thought I should have certain responsibility to do this because I know the subject, understand better than many non-biologists, right? So I could communicate this widely and also actually do some work, And the second part is cancer biology, which I'm doing now. I was interested in understanding the how specific organs attain a specific size given the proportionate to the body size of a given species. For example, in human, the two
mm,
are exactly the same size, but the size of
mm,
our is proportional to body size. That's how that species specific features are embedded in
mm,
our, you know, what called as size and forms, right?
mhm,
Or
mhm,
form and size. So that question, when I was asking, it turned out to be that a lot of this is related to growth control, variety of different molecular mechanism involved in growth
mm,
control.
mm,
As
mm,
a community of cells, a large number of cells, it could be millions of cells, would divide in a synchronous way. And
mm,
one or two, if they don't compromise in a synchronized way, they actually get killed or competed out and so forth.
mm, mm,
In spite
mm,
of this thing, cancer happens, and cancer is, basically growth control gone wrong.
mm,
But the
mm,
same mechanism, right?
mhm, mhm,
Growth mechanism. So that's how it, naturally transitioned from Drosophila development biology to human cancer biology.
mhm,
And that
mhm,
basically, and then the clinicians, when I was talking to them, they asked some interesting questions, which is also socially relevant questions in the context of health. So
mhm,
then I said, oh, these questions are more important for clinicians, but they don't have the tools,
mhm,
the way of addressing these
mhm,
questions. You need certain expertise, certain methods of doing designing experiments, and actually
mhm,
tool resources. So that's how I got into clinical research now, right? So you can see the transition. It's simply because whatever you're doing has an implication and has an impact in other fields, and there was an opportunity was
mhm,
there. Not that I was
mhm.
going after that one. Somehow these opportunities came in front of me, and then I grabbed it.
I see. Fantastic. So you mentioned, about the organ development and the size comparison. So, for those who are not biologists and also attending your podcast today, so for the listeners, uh, can you explain how can such a tiny insect help us to understand the human body build?
Sorry, repeat the question. What do you have to explain to whom?
So basically, I mean, we would also have a lot of non-biologist
Oh, I see.
speaker
Sorry. Okay. I hope I
audiences,
didn't use too many jargons. Yeah. Tell me. Yeah. Ask me.
So no, I'm definitely sure our participants would love it. But especially, we all study what is model organism in general. But how Drosophila makes it different, especially, and how can you study human diseases
Oh, yeah,
in the
yeah.
model organism?
So when you want to study diseases using flyer model or any model like a mouse one,
Mm-hmm.
right? It's not that how different they are. You ask the question
Mm-hmm.
how similar they are, because finally you want to model how we grow, how we develop,
Mm-hmm.
or how our
Mm-hmm.
diseases are manifested. Differences are there.
Mm-hmm.
It's very obvious, right? A fly is fly, human is human, right? A genie, right? So differences are
Mm-hmm.
obvious. It is the similarity that you
Mm-hmm.
draw, right? I'll give
Mm-hmm.
you two example, which is most common sensical example, which in a school textbook one can tell you. All living
Mm-hmm.
forms today have exactly the same, including all the fossil records so far, what we know. The chemistry of life
Mm-hmm.
is exactly the same across all
Mm-hmm.
organisms, bacteria to human,
Mm-hmm.
right? It's a DNA-based genetic material, sometimes RNA-based in some viruses, but don't
Mm-hmm.
worry, viruses. And DNA to RNA, RNA to protein, this the mechanical mechanism of all of this
Mm-hmm.
happens. Fidelity of
Mm-hmm.
information transfer is the whole mechanism is identical to an extent that take, let's say, DNA piece coding for, let's say, human protein, let's say insulin, put it in bacteria or yeast, you can make human
Mm-hmm.
insulin, right? Because when you put it in bacteria, they also consider simply a polymer of nucleotides, the DNA.
Mm-hmm.
And the chemistry of DNA replication, DNA, RNA transcription, or protein translation is
Mm-hmm.
exactly the same. So what comes out is based on the what sequence you have in the DNA. say insulin
mm,
protein or some other XYZ
mm,
protein, which normally encoded
mm,
in bacterial DNA, right? So this is how we actually use any other model organism to study human diseases at the
mm,
molecular. Now, similarly, when it comes to development, right? Now, from a single cell embryo, multiple cells put through cell division,
mm,
the head side and which is the tail side, and where different
mm, mm,
parts of the body get what called one side of axis is the the head to toe axis, we call anterior to posterior axis. Second is what's
mm,
called front to back axis, we call as dorsal to ventral axis. Now
mm,
these mechanisms of these highly conserved mechanism, right?
mm,
What
mm,
during evolution, for example, chemistry of DNA or chemistry of life has not changed in 3.5 billion. The living system will die. It's not any more living system. So nothing happens. They will eight cells and how they get organized, which order they get become head versus toe versus front versus back.
mm,
All of these are highly
mm,
conserved. Then
mm,
additional, mechanisms evolve during evolution. One become fly, one may become something else, kind of a thing. As more and more
mm,
complexity, or you have vertebrates. Among the vertebrates, you have multiple different forms of vertebrates, and including mammals at the later stage of evolution. And amongst the mammals, we have humans. You can actually see that quite a bit of these mechanisms are highly conserved, but it doesn't make that a fly can address all human biology or all diseases of human.
mm,
It's only
mm,
certain aspect
mm,
of it you can study. In cancer, for example,
mm,
if what are the growth, how multiple cells together can synchronously divide, you can study
mm, mm,
cancer. But what can go wrong in this mechanism, which is conserved, you can study in human. Mistakes into Drosophila and make cancer into Drosophila, generate cancer into
mm,
study that one.
mm,
That's what also we have
mm,
done. I hope it
mm.
was enough jargons.
You nicely connect, developmental biology with cancer, and also a central theme of your work is understanding how organs, grow, end up in the right place, and also they develop right size and shape. So, firstly, why these questions are important or fundamental to answer in understanding biology, and at the same point, why do you think that sometimes, as in cancer, it becomes so difficult to control the division of the cells, or rather, how would you connect as a developmental biologist to cancer?
So I'll tell you what. So the first part you were telling about the model organism. The
Mm,
life is, as you know, irrespective of how much we know, how simple even the chemistry of life, the at least fundamentals
mm,
of DNA, the genetic material, how information is coded or decoded, and that this
mm,
whole process is so elegant, and you can always say
mhm,
it's very simple, but life is, it's very complex, particularly
mhm,
multi life is even more complex. And if you look at the
mhm,
way we adapt to, in given our own lifetime, varying, you know,
mhm,
environmental and other conditions, and then how over long term, generationally, we adapt to changing environment and how we evolve.
mhm,
All of this is
mhm,
extremely fascinating, but it's also complex. To understand such complex phenomena, no one individual can work on. Right? We need
mhm,
a community of scientists to work on it. Across the globe, people are working on it. Now, how do you compare the
mhm,
results of mine with someone else? Right? So you have to have an apple-to-apple comparison. So that if you go deeper and deeper to understand life, you need to have multiple inputs coming from different scientists having researched on the similar topic.
mhm,
And then
mhm,
if I say I worked on flies, someone else work on, let's say butterfly, someone else work on a mouse. Each one work on different aspects of life, and then see, can you build a whole life together? It's very difficult.
mhm,
So that's why consciously scientists decided, let's use what's called model organisms. You go deeper and deeper in bacteria, E. coli, yeast, Saccharomyces, or Drosophila, or Arabidopsis as a plant model. So if
mhm,
dozens of people work on the same organism, right, across several years, and you go deeper and deeper on very different aspects of life. And then when put together a story, it will be much more comprehensive. You get much deeper insights to life. It's exactly what happened, right? If you look at the fly biology is more than 100-year-old now, and
mm,
millions of people have worked on it. And what we know about
mm,
is largely come from such model organism, equally yeast, Drosophila, and so forth.
mm,
So that's the
mm,
purpose of model organism, and it makes sense in evolutionary context too, because what is true for Drosophila could be true for human too, with a pinch of salt.
mm,
Now, coming to your question about the cancer part, see, as I told you, if a development is, we all are born as a single cell first. Egg and the
mm,
sperm that coming from the parents get fertilized, and that fertilized cell is one cell. And our
mm,
body is made up of 50 trillion cells. House
mm,
fly is made up of few million cells. Now, if you have to get a 50 trillion cell complex body with so much distinct cell types and tissues and organs, how do you get from a single cell? So there is a series of events. In these series of events, two things will happen. One is one cell to you get large number of cells, like 50 trillion cells over a few months. At the same time, a group of cells attain specific identities. Something become head, within the head, something part of the brain, part of the eye, part of the ear, part of the mouth parts, and so forth, and nose. Similarly, there are multiple such tissues and organs and internal tissues of muscles, neurons will develop. So given all of these uh, processes, right, what you really see is there is a lot of collective growth, collective interaction happens. Otherwise, you cannot get one body with physiological connections, which will function as one unit. If 50 trillion cells, not looking at 50 trillion individual cells, they collectively become one. So there is a lot of interdependence, interconnectedness, and synchronization happens early during development to ensure that this is what happens later. This is the outcome. And that synchronization, if it is lost, you get all kinds of growth problems. Sometimes overgrowth, sometimes stunted growth. Right? Sometimes completely defective growth, nothing happens. Finally, the embryo or fetus dies. So in this context, the connecting to the cancer is the developmental events are recapitulated in stem cells, which we are known as stem cell, because we live longer. Where we live longer? Because we have the capability to, we repair wear and tear of our tissues, worn out tissues, or repair if there is an injury, a wound healing. And these are all done by specialized cells called stem cells. They are kept aside in our body during development. They don't contribute to actual building of whatever the tissues and organs we already have, but they are kept aside and remain to repair if there's a wear and tear of the tissues, like heart. For example, heart has to beat for 24/7 for, let's say, 100 years. You can't expect the same heart cell will function like this. So it will periodically get replaced. You will actually have a multiple times new heart, but bit by bit, new heart is built. So you will never realize that actually during the birth and the death, the heart is so different. Similarly, liver, lungs, your gastrointestinal and everything. And skin layers, we get multiple times, new layers of skin. All of these things happens because of stem cells, including, of course, wound healing. And those stem cells have to divide again synchronously against a specific cue or a signal or a trigger. If they start dividing on their own, they become cancerous. But how they are controlled for cell division to repair a tissue which is exactly the same how it originally developed that tissue, right? Heart or liver or skin, whatever. So that's why development biology, stem cell biology, and cancer biology are completely, basically one aspect of biology, growth biology you can call, but they are all
mm,
three, sub
mm,
branches of biology which basically are connected. And so it's easy to work on cancer if you're a development biologist, easy to work,
mm,
cancer if you're a stem cell biologist or a developmental biologist. So the three are always interlinked. We know a lot about very different genes involved in growth control during development because cancer
mm,
biologists identify them first. And with cancer biologists know quite a few aspects of cancer biology because development biology discovered that first early during development. So it's by
mm,
direct discovery.
mm. I really find this fascinating how a developmental biologist connects cancer, because when we directly talk to a cancer biologist, their thoughts are different. And especially when you talk to someone who is in the
Medical.
field of cell death, explains cancer completely in a different way. So I really love, as you correctly mentioned, where there is an amalgamation of, not just one discovery, but multiple discoveries and pathways, and then leading us to,
Okay.
some discovery and knowledge. So this is something that I would really love. So looking ahead, what are the big unanswered questions that you would love to solve, or you would love to see them solving, in evolution of your own research or your own field?
So I have reached a stage where obviously I'm not going to solve anything more. I'm almost at the end of my I mean, I'm not that I'm not going to be inactive, because at a stage where you know
Mm,
I'm more facilitator of others doing research, doing my own research, but I
yeah,
still have my
yeah.
own biases, my own personal interest.
Mm,
One is I'm very fond of uh, two aspects of biology. One is the very beginning, you can call as origin of life. I really want more and more people in India to work on origin of life. Globally also, there are
mm,
very people, I want, of course, it's science is all about humanity. So
mm,
I want more and more people work on origin of life across the world.
mm,
But what we know so much about the biology is what has already evolved over long time.
mm,
What
mm,
evolves is what started evolving is something which started
mhm,
5 billion years ago. But that point is still know um, less known. I mean, in fact, hardly anything is known about that part. The
mm,
origin of life is the
mm,
most unanswered question in biology. Second is the human cognitive neuroscience, because
mhm,
there's so much of misconception about ourselves, what we are, who we are, our society, and everything, is because we always put ourselves in in the center of the whole universe, and then we dominate,
mm,
thinking that we the ones who are driving everything, but which is not true. The neurobiologists and cognitive neuroscience people have actually again and again emphasized how your the past memory, past experiences, what new stimuli
mm,
you get through your sensory organs, analyzed in the brain, and the brain comes out with certain kinds of behavioral response, and that behavioral response,
mm,
certain analytical framework, is also an evolved phenomena that evolution is all
mm,
about. The selection evolution is about survival and procreation, your ability to produce
mm,
next generation. So the fitness, what we
mm,
call evolution, is what drives what gets selected, right? And what
mm,
gets
mm,
selected is already selected. Is that in that framework only our brain functions. Unnecessarily, we
mm,
think too much about ourselves, and we almost think
mm,
that we are the gods, or if there is a god and we are the direct descendants of the god, and so forth, right?
mm,
So
mm,
that's
mm,
something which we need to properly address this problem, and come out, because it
mm.
also not only social harmony, it also has so much of neurocognitive diseases and so forth, right?
Absolutely.
Third most important part we need to understand is in the context of we so understand the evolution of the modern biodiversity that we see today, right? And the current one,
Mm,
whatever is extant, the
mm,
species, and this all so rich biodiversity. Thankfully, we still have good biodiversity although it's small parts of the world. It could be Amazon, it could be Western Ghats, or in the Northeast in India, and so forth.
mm,
But
mm,
this biodiversity is in a fine balance, what call ecological balance, right? That
mm,
role from bacteria to all the way to large mammals, they have everybody
mhm,
has to play, and that role,
mhm,
we understand the importance of
mm,
these relations and
mm,
behavioral responses, or chemical response and so forth, but that some of the fine ecological mechanism is not that understood.
mhm, mhm,
So understanding the ecology and biodiversity in greater detail is another area which more and more people should work on it. That
mhm,
helps develop new conservative policies and strategies,
mhm,
right? Without simply they will want to conserve this will not work. Anything,
mhm,
the
mhm,
tiger or lion or an elephant is part of an ecosystem. You can't conserve
mm,
individual species. You have to conserve a whole ecosystem.
mm, mm,
So I think ecology and biodiversity is going to be another area in which I'm really passionate about and
mhm,
trying to facilitate that part of the work in the country. And
mm,
also helps in, for environmental protection to biodiversity
mm,
conservation and then overall sustainable living.
mhm. I see. So, this was a really nice, a direction as well, where leading institutes of India are also currently facing and, making their research orientations as well. So, what do you think? What does building or managing a scientific institution mean to you beyond just, laboratories and science?
Yeah. So for me, it's not a routine administrative thing. That's what I consider this. For me, it's an exciting role because there are such fantastic scientists. In fact, I always thought there's so many good scientists. Do I really make a mark in my own way to do science because there are
Mhm,
such fantastic scientists, right? In fact,
mhm.
if you read the referees' comments or manuscripts, we'll know how poor scientists we are many times. Your referees come out with some fantastic insights, which we would have forgotten,
Mm,
we would not have seen in our own data, right? Of
mm,
course,
mm,
there's always a mixed responses, mixed experience. All of us have, you all have that. Sometimes referees can be too caustic. Some referees may not have read the paper. It's there in the paper, but they say, what about this and kind of stuff. But that's okay. There's always
mm,
small percentage of referees. Generally, good referees give good comments. So knowing all of these things, I always thought I should, be a facilitator of doing research rather than doing my own research. While I need to do my own research to keep my passion intact and be always excited about science. But
mm,
I almost in mid-career time, within 10 years of doing research, independent research in
mm,
CCMB in Hyderabad, I realized that, I can be more productive by helping others to do research. Say, for example, if
mm,
I contribute X unit of, research, with me and with my own students, with all the resource
mm,
mobilization activities and other things,
mm,
if I can help 10 other people and the contribution is 2x or 3x or 4x,
mm,
it's
mm,
so finally science benefits, community benefits, humanity benefits, and so forth. So in that context, I started actually taking up the role of science administrator right from
mm,
early my life and career
mm,
life. And I
mm,
really, because talking to young scientists, even senior mid-career scientists, it doesn't matter who they are, they come with such good ideas. Most of the time I don't understand anything what they speak because for example, if someone comes from biophysics or physics background or mathematics background or computational background, or working on origin of life or biodiversity, I don't understand most of what they speak. But I know, what they talk about is high science, good quality science. They are so excited. So if
mm,
they say, "No, I really need some money or need some resources to do this, can you help to get some permission?" I
mm,
I get excited to that part, which is mundane administrative part in a way. But for me,
mm,
the excitement I see in their
mm,
eyes and eyes and face.
mm,
That's what
mm.
keeps.
So, apart from science, do you also have some hobbies that you would love to do when you are alone or during for your own time?
Yeah, I used to have, always used to read literature. That has come down a lot.
Mm-hmm.
They don't get time.
Mm,
One
mm,
thing I do is
mm,
to, of course, listen to music, Indian classical music,
mm,
particularly
mm,
Hindustani classical music, which what I,
mm,
appreciate most. Although I'm not an
mm,
expert in any of that one, but I can listen to music for hours without any problem. So, I mean, without getting bored. So, anyway, classical music is something we don't call it as a hobby because hobby means you actually invest a lot of time and actually pursue as like a parallel, part of your.
mm,
I
mm,
don't overly spend time on these things. For example, if you are in a flight where you don't have anything else to do, you keep listen to music. If you're going for a walk, you listen to music. But I don't
mm,
use it as a parallel uh,
mm, mm.
activity of
I
my life.
see.
Yeah.
Hmm. Something which goes on with you. So, I mean, there are many questions that we would love to ask. Our team was preparing. We also get some from the audience. So the questions are like amalgamation of, mixture of a lot of things going from personal to professional lives and driving everywhere. But something I would love to end this podcast before I take students question, especially from YouTube. uh, what advices would you like to give to our audiences, and especially the young researchers, today, not having, I mean, not having that much amount of patience over the time, with the running technology and dopamine everywhere. What suggestions or
Okay.
advices you like?
I can't give a very general advice to anybody because I'm not such a uh, senior person. But as
Mhm.
individuals, yeah, I'll tell you, most of the time, people get frustrated because you don't get money to do the kind of research you already thought you would do it. So you go as a PhD student, then you're as a postdoc, you want to start your own, research. And they say that without this confocal, without this, mass spec, or without this kind of you know, a tissue culture room or clean room, I can't do research, and they get frustrated. Or they'll say, I don't
Mm,
have to do research, and they get frustrated. So my suggestion, this is exactly what I've been doing myself, and also advising everybody whom they come
mhm,
talk to me. There's so
mm,
much one can do in research. There is so much, low cost research also one can do. And today,
mm,
data is available online. Everybody, whatever the papers have already published and people have put up data, whether it's
mhm,
data
mhm,
or spec data or simply epidemiological data or biodiversity,
mm,
ecology data. Every
mm,
manuscript only is scratching the surface, that data. That data is still a gold mine. Which are published, it could be Nature paper or it could be another smaller journal. It doesn't matter. It's
mm,
everybody would have only scratched the surface. So one can actually sit on a computer and can do fantastic research and come out like TCG in cancer biology. People have can
mm,
easily publish only doing the analysis of the data is already there in TCGA.
mm, mm,
Similarly, there's another low-cost research. You can do ecology, biodiversity. High quality research is what one should aim for, and not for simply publishing papers. High quality research
mm,
can be with low resource setting too, and people can get continuously be excited about doing good research if the
mm,
quality is good. Don't worry where you publish it, and how good quality is. See, when you ask, is my work how high quality? More than the outsiders, your inner self will be able to tell you, because the process by which you went through that research, how you discovered something, how you wrote the manuscript, how you interpreted the data, it makes you feel that AI is of high quality. If you think it's high quality, genuinely, it
mm,
is. It's always high quality. You don't need another person to come and tell you it's of high quality.
mm,
I mean,
mm,
you may make mistakes. You may not be able to accept others, because accepting others
mhm,
is how well you communicate. So that
mhm,
communication skills we have to develop anyway. But
mm,
quality, you would know yourself. You don't have to ask another person.
mm. So
You
basically, you don't need others' validation to, tell your research that how good it is. It's rather you yourself
Yeah, yeah.
would drive it with curiosity. I see.
So,
So normally I ask one question to many scientists or people whom we have hosted that we admire, and we have hosted some couple of scientists who are also are from those eras when there were no computers, and now they see how it's going. So one thing that I would like to ask you is, what is your favorite scientific discovery in the science so far? It could be like few PIs, they gave answer as discovery of H plus ions and Cl minus ions, something like this. So what discovery fascinates you? There could be multiple.
I mean, I told you, right? Doing research itself is should be exciting, right? Designing an
Mm,
experiment.
mm,
Discoveries. every discovery is important. The discovery
mm,
means, basically something which is unknown, right?
mm,
Translational or applied is what is known. How do you, how you solve a
mm,
real life problem, right?
mm,
Even that also is a process of discovery, because taking the path of knowledge
mm,
to solving a real life problem, there's also lots of gaps, right? And
mm,
finding the real life problem which is addressable, solvable, and then see whether there is which knowledge will correlate to which real life question which you can translate and everything. So discovery
mm,
happens in everywhere, right? So the
mm,
something which is not known to others that you have found out, and then you will be able to convince yourself, and then you will be able to convince others through a good communication skill.
mm.
And of course, scientific methods is itself is most exciting.
I see. I see. So, since you mentioned about growing age and also how well science is moving ahead, what do you expect from upcoming generation, or what contributions you see coming from the science?
Okay, so I'll tell you what. Most important first part is, gone are the days where individuals have to think about what I can contribute individually, and using
Mm,
only my name as a prominent discoverer or a
mm,
provider of answer to scientific questions. I think
mm,
more and more we are moving into team science in this world,
mm,
because of the modern technology of connecting the world over internet in real time, and we're going to address the many big problems, questions,
mm, mm,
collectively rather than individually. Science has always been a collective endeavor, but it is not necessarily in real time, right? Without
mm,
Galileo and Kepler, Newton could not have achieved, or without them, Einstein could not have come out of nothing. So finally, everybody is standing on the shoulders of other giants. But that was
mm,
happening on different times points. So
mm,
shoulders were somewhere else, and some other time scale, someone would be standing on the same shoulders, kind of thing. But today, it can happen in real time. That's how COVID, for example,
mm,
everybody shared their discoveries and understanding and interpretation very quickly, and collectively we could address and come out with a vaccine and so forth. So I
mm,
guess more and more team science, collective science will come out in future, and
mm,
we in all areas of science. And so people have to, you know follow scientific methods which high levels of ethics and integrity, right? There is no way one can get away with any kind of falsehood, because science has always stood the withstand time, uh, the test of time. And that's because it has a self-correcting mechanism. The process of science, the methods of science, has a self-correcting mechanism. You can be
mm,
unintentionally make mistakes. You may have been in your interpretation, but science corrects itself, right, over time. But that is accelerated now because of the
mm,
way so many people are doing it. So falsehood is also discovered very soon. So no one is going to win by falsehood. No one is going to
mm,
win by shortcuts. Scientific methods had to
mm,
be followed, right? Shortcut is bypassing the scientific methods. But acceleration is different. Acceleration is because of modern technology can accelerate the discovery, but
mm,
not
mm. I see.
So that's my suggestion. And what's problem going to solve? Bring scientific temperament. Hopefully, more and more people understand that there's hardly any difference between different races, castes,
Mm,
religions. Which people are always using this to fight among themselves. And it's hardly
mm,
any. We are only 250-year-old, 250,000-year-old species on Earth, whereas
mm,
life is on Earth is 3.5 billion year old, and there are so
mm,
many mechanisms which have evolved to live at you know at the minus 60 degrees centigrade to 120 degrees centigrade in all
mm,
kind of geoclimatic conditions, and
mm,
each one have their own ability and the limitations, and we are also one
mm.
such species, and trying to use us fantastic,
mm,
statement, very, very practical and philosophical. Is when you look at the whole Earth, starting from a high, you think all other, difference of opinion, arguments, and these differences, conflicts, it becomes completely, you know, non-consequential, completely
mm,
no importance in life. Just the whole Earth is just one, piece, and it's living for 5 billion years, and then we are
mm,
simply fighting amongst ourselves, for no reason. Right? I think that's the life we have to lead in the society, and science helps. No other,
mm,
you know, no other, area of human activity can help this.
mm. I see. I think I really enjoyed, this candid conversation with you, and I mean, it would have been best if we could have, made this over a cup of tea or a coffee
Well,
with a
I'm having my
podcast.
coffee. Yeah.
So absolutely. So before ending the podcast, I would really love Somilia Mishra to take up some audience questions that we have from the registered audience. We have many questions from
The audience they have asked, they have a lot of questions, but I'll take only a few couple of questions. The first questions they have asked is that what is the role of AI and ML in studying embryology, and what is your perspective on it?
Say it again. Repeat. With two specific question. Yeah. What is that?
Sir, actually, the people have asked that how AI and machine learning is helping in understand embryology and developmental biology. That the people have.
So, oh, yeah. So, no, no, there's hardly any difference, right? Embryology is the study of embryos, but typically when you call something embryo in zoology, the word embryo is used. In human, we call the word fetus in medical terminology. And but starting from a single cell until the free living organism emerges, we basically call as that entire area is called development biology. Earlier, they used to call as embryology. It's a modern, so, terminology is more developmental biology because it's not just looking at embryo, because sometimes it goes beyond embryo, right? So, for example, in case of insects, let's say, certain kinds of insect like silkworm or butterflies, a free living is a caterpillar or a larva, right? And then again, further development happens in the whole adult fly emerges, which either butterfly or a fly, right? So that entire process is developmental biology. But if you say embryology, you would stop when the caterpillar is born. Then you stop, but actually development continues beyond that. Similarly, in human, developmental biology is not just, if you say embryology or fetus biology, within 3 months everything is done. There'll be, it'll be kicking its, legs, and heart would be beating in a few months time. But it goes beyond that one, and beyond the birth also, postnatal, what you call. So development biology is sort of in a much more comprehensive.
Yeah. So, sir, second question is that, sir, can genome card like soil health card will be available in future to work out diseases to be controlled or prevented?
Say it again.
Sir, can genome card like soil health card will be available in future to work out diseases to be controlled or prevented?
Yeah, yeah, it's already happening. So gene therapy or gene editing and genome editing is already in the experimental stage. It's happening for the last 10, 15 years in the lab,
mhm,
whether you correct the gene in the mouse or a fly or in other ones. But clinically also people have now already done it. India has recently last year published, Alok Srivastava from CMC Vellore, he published where he treated 6 thalassemia patients, beta thalassemia patients with gene therapy. All 6 worked successfully. It's a fantastic achievement. And more such will happen. mRNA therapy, gene editing or genome editing. Only thing is one has to be very careful because we should not overdo things because it can have other consequences, right? Those individuals, when you correct it, will be young people and they have to live longer and then also have their own children later. So it should not have an impact of their own survival and long further life, or at the same time, it should not affect the next generation, right? And then whole population and kind of a thing. So I think one has to have a clear understanding of the cause of the disease, how the disease progresses, what are the other impacts of disease, and when you correct, what are you correcting comprehensively, or correcting only the symptoms or only one small aspect of the disease. All of these things have to be understood very clearly before you go for gene therapy. Because one of the things happens is what we would call as many genes are called pleiotropic. They have certain role in early during development, may have a slightly different role later during development at the physiological level. So these are the things one has to look into it.
Yes, sir. The last question which I would like to ask, which is very important for us as well, that as a researcher, how you managed your failure? Because in life we face a lot of failures, and it is important to get managed to it. Then how did you manage?
Well, again, science helps, right? And if you are truly a follower of scientific methods, and as you know, scientific methods is that every experiment, the chance that it will be successful is actually very low, right? You should be surprised that your experiments work in the first round itself, right?
Yeah.
And that basically helps you to say, okay, yeah, manuscripts get rejected, you improve the work, and then you get accepted at some point. You may not be the first one to publish, but you still publish what you have published in some difference to others, how they have done it. And also, if you just whatever you're publishing is already, let's say someone else has worked, but you didn't know, or it happened that they published ahead of you by a few months. But the science progress only by further validation and verification. In a way, if two groups, two different parts of the world independently working converge into the same results, it's good for science. It may not be good for your career because you may think that you're only secondary to someone else. But the scientific community has to understand, particularly those who are involved in promotion and other things, that as long as someone has worked independently of others, it still should be considered as an important piece of work. Anyway, but in terms of failures, failures in other parts of your life also, you can take it easily without much, depression or anxiety, as long as in that's how the life is all about. Because that science helps you to learn that.
I see.
Yes, thank you, sir.
So,
Mr. Noor. Thank you for so much for addressing.
Thank you so much. I really enjoyed talking to all. It's a very good interaction. It's the first time I'm actually getting these kind of questions. So I'm really,
I hope
really
we
enjoying
would
it. In fact,
get
I'm already.
much more better from today with evolution, and I mean with evolving of of the thing. So as we come to close the today's session, I would like to take a moment to express me and also all our team's heartfelt gratitude. First and foremost, sincere thanks to you, Professor Shashidhara, for joining us, taking some time out, sharing your thoughts and experiences, especially over the time, which truly were inspiring, gave an enriching and complete candid conversation today. Deeply grateful that you accepted
Thank
our
you
invitation.
so much. Thank you so much. Yeah.
I would like to also thank our wonderful audience, who would be engaging in future, and as well the curious group that we have always connected. Thank you so much for your enthusiasm. So we would post like feedback form on the chat box, and please give us some feedbacks from there so we can improve, get good speakers for everyone. Big thanks to entire team of the SciComm Synapse organizers, volunteers, everyone behind the scenes who makes this possible. So we wrap up today's episode. Thank you all for being there, and we hope to see you in future events. Thank you so
Thank you
much.
all. Bye, everyone.
Thank you
Thank
so
you.
much. Bye. So for all the audiences, there would be a feedback form in your YouTube channel. Please check it. Please fill the form to get your e-certificates. Please fill up the form, and also it helps to get better, get more speakers, and upcoming podcast. We are getting fantastic scientists across India now, and it would be fun. Hope to see you all again.